Okay, today's bell ringer, day 56, says hornless cattle, big H, is dominant over horned cattle, little h. A homozygous hornless bull is mated with a homozygous horned cow. A homo means the same. So if it's homozygous hornless is dominant, so it's big H. Homo means they're going to be the same. So for our homozygous hornless bull, its genotype is going to be big H, big H. Okay, that would be our bull. Okay, and then it says, it's mated with a homozygous horned cow. Well, horns are recessive in this case. Homozygous means both alleles are the same. So the cow is going to be little h, little h. Okay. And then we have to make a Punnett square and put these in to see what the offspring are going to look like. So we're going to attempt to do that. Let's insert a drawing. Okay, so we draw a Punnett square, and then our bull was homozygous dominant, so he was two capital H's. And we're going to need scribble. This is going to be big H, big H. And our cow was homozygous recessive, so she will be little h. Little h. Okay, then we just fill those in, bring our big H down from here, and just to make it look consistent, look nice, if there's a big letter and a little letter, you're usually going to write the big letter first. So we have big H, and then bring our little h across. We have little h. Oh, that's big H. And then over here, we'll bring this big H down. And this little h will come across. And on this one, we're going to bring our big H down. And our little h across. Our big H down. And our little h across. Okay. So now we have all four of our potential offspring here are big H, little h. Okay, we said homo means the same, so if they're homozygous, they have two alleles. They're the same. They got a dominant allele from mom and a dominant allele from dad, or a recessive allele from mom and a recessive allele from dad. They have either two recessive or two dominant. If they're homozygous dominant or homozygous recessive, the alleles are the same. Well, notice all of these offspring ended up having a big H, little h. Big H, little h, big H, little h, big H, and little h. That's what we call heterozygous. Hetero means different. These each got a big H from the bull and a little h from the cow. Because the bull only had big H's to give, cow only had little h's to give. So they ended up with 100% of their offspring being heterozygous. These are homozygous. These are heterozygous. Hetero means different, so they have two different alleles. They have a dominant allele and a recessive allele each. So the dominant trait will be what will show up. Okay, so next it says, what will be the genotype and the phenotype percent of the offspring? Show your work. So we just did our Punnett square. Genotype has gene in there. So genotype is looking at those alleles, those genes. What gene did you get from mama? What gene did you get from daddy for that trait? So the genotype is looking at the genes. Okay. Phenotype, pheno, even though it's a pH stuff like pheno is those physical traits. Physical traits. So pheno starts with pH, physical starts with pH. The phenotype is the physical trait that you're going to be able to observe based on the genotype. So you have the genes that you got from the parents. The H's came, those are the alleles, those are the genes. Got a dominant gene. Dominant gene came from the bull. A recessive gene came from the cow. So that's the genotype, big H, little h, for each of the offspring. That would be genotype. We'll put genotype. And genotype, it's got gene in there, so it's looking at the genes, the alleles that make up those. So genotype is big H, little h for each of the offspring. Okay. Phenotype. Pheno is looking at those physical traits. So phenotype for each of those offspring is going to be, and we have big H, little h. Big H is dominant, so that dominant trait up here told us that big H was hornless, and it was the dominant trait. So if you have an allele for that dominant trait is what's going to show up. That's what makes it dominant. Even though there's an allele there for the recessive trait of being horned, the recessive trait will not show up in the presence of the dominant trait. Okay? This is the law of dominance, that even though it's heterozygous, or if you have a parent that's homozygous for a trait, that all of those offspring are going to show that dominant trait. Law of dominance. So the phenotype is they're all going to show that dominant trait of being hornless. The phenotype is hornless. Okay. So that's Punnett square, just simple dominant recessive alleles. Next it says describe nondisjunction and when it occurs in meiosis. So we talked a little bit about this earlier this week. Nondisjunction is when the sister chromatids fail to separate appropriately. So nondisjunction is when the sister chromatids fail to separate appropriately during anaphase 2. Of meiosis. Okay. So that's when you have the little X's and you're at metaphase or lined up down the middle, single file during metaphase 2. We've already separated our homologous chromosome pairs. So now we're down to just our single X's there for each chromosome set. So we have they're lined up down, single file down the middle. During anaphase 2, those sister chromatids should separate and pull apart. One going to one side of the cell, the other one going to the other side to make two complete sets of chromosomes to make two new cells. Well, if they fail to separate appropriately, they don't come apart at the centromere, one going to the other side, one go to one side, one go to the other. They fail to separate appropriately, then that's what's called nondisjunction. So it's when the sister chromatids fail to separate appropriately during anaphase 2 of meiosis. It results in gametes having too many or too few chromosomes. So half of the one that got the extra one would have too many. The one on the other side would have too few because it did not get its is chromosome from that sister chromatid from that chromosome. So it results in gametes having too many chromosomes or too few chromosomes. And we talked a little bit about how Down syndrome is the result of non-disjunction, along with several other disorders, but Down syndrome is probably one that more people are familiar with. And it results when chromosome 21 in either the egg or the sperm had not separated, so either the egg or the sperm had 2 copies of that chromosome 21, and then when it gets fertilized, that makes 3 copies of chromosome 21, so that's called trisomy 21 or Down syndrome. And that's the result of non-disjunction. And the next is, what is synapsis and when does it occur during meiosis? Synapsis is when those homologous chromosomes pair up. They pair up and form what's called a tetrad. Remember, tetra means 4. So the chromosome number 1 from mama now has 2 sister chromatids because DNA has replicated. Chromosome number 1 from dad now has 2 sister chromatids, so 2 mama chromatids, 2 daddy chromatids together. That's 4 chromatids for that chromosome at that point. It's called a tetrad. So synapsis is when those homologous chromosomes pair up and form what's called a tetrad, allowing crossing over to occur. Remember, crossing over is when those sister chromatids kind of wrap around each other and exchange some genetic information, and it's what adds to genetic diversity. It's each time it happens, it can happen in different places, and that swapping of genetic information doesn't happen the same way twice, so that adds to genetic diversity. It's what lets you look like your siblings, somewhat like your siblings, but not identical to your siblings, even though you have the same mom and dad. So that adds to the genetic diversity. That crossing over happens during prophase 1. So it happens right off the bat. It happens during prophase 1. And it adds to genetic diversity. We also talked a little bit about random assortment, how one egg is not going to get all the mama chromosomes, and the other egg gets all the daddy chromosomes, or whatever. They randomly assort. You may have 2 from mama and 44, or 2 from mama and 21 from dad, or something. Or you may end up with, I don't know, 10 and 11 due to random assortment. And that random assortment would happen as they line up in the pairs. They can be on either side. It's not going to be all one side mama, all one side dad. So that's another thing that adds to the genetic diversity. Crossing over adds to genetic diversity, and then the random assortment of those chromosomes as they line up also adds to genetic diversity. And that's that for our well rigor today. It's kind of long, but there was a lot of information there. And there it is again if you need to get some of that. There's that one. There's that one. And there's that. And again, you can go back and scroll back up and look at it again if you need to. So there's it.